we have the honor today to welcome Kathryn Gronlund from biology, and her title today is Epigenetics, Where Nature Meets Nurture.
Right, thanks for asking me, I appreciate it. So how many of you remember the year 2000 when they released the preliminary sequence of the human genome? Yeah. And the year 2003 when Craig Venter and Francis Collins did their joint press release and said that the Human Genome Project was complete. You guys remember that? Yeah. Oh, yeah. Okay, so momentous stuff for biologists, right? But in fact, I've got the original copy of the paper releasing it. It is signed by Francis Collins. For me, though, a couple of weeks after that, I heard an interview with a man by the name of Randy Journal from Duke University. And he said, well, that was a good start, and now the real work begins. And it was like, what? You know, is he dissing my hero, Francis? And so I looked up Randy Journal, and it turns out that he was born and raised right down the road from where I was born and raised, and that he wrote prolifically about cancer biology and about epigenetics. And I've kind of been hooked on epigenetics ever since then and following all of that, and so I thought you guys might be interested in it also. So before we actually get into the epigenetics, I just want to do a real brief DNA science thing just for a couple of minutes. So you guys, I'm sure, have all seen the double helix, and so the important parts here is that we do have the base pairing in the middle, A's T's, C's G's. It's going to be these C's that are going to end up being very important to us a little bit later on in this discussion. And so DNA has two big, huge purposes. One of them is to make sure that the genetic information is passed on to the next cells. Thank you. So before cells divide, they're always going to first replicate their DNA so that the new cells can have pieces of the same genetic information. And so we do that by two processes. This talk is mostly going to be about multicellular eukaryotic organisms. And so we do that by two processes. One of those processes is mitosis, and so you're going to be making more body cells, repair or growth, replacement of old cells. And the other process is called meiosis, and what you're doing is making eggs and sperm cell. We'll talk about that in just a little while. And so the other big function of our DNA is to give the instructions for making your proteins and your RNA. And so we do that by a process called gene expression or protein synthesis. And so you're going to take the DNA, there's going to be a section that's called the gene. You're going to open it, you're going to make a copy of the instructions of that gene, and then you're going to go ahead and you're going to build the protein. And the protein is generally what gives you your phenotype, your traits, your physical characteristics is your phenotype. It's caused by your proteins. So those are the two big things that DNA do. And you probably knew that, but one of the things that non-biologists don't know is sometimes is that DNA coils a lot in order to get very compact to be in your cells. And so the DNA isn't hanging out in your cells right now in its strung out form like that. It's going to be coiled around proteins, and so the proteins that it's coiled around are called histones. And so it can get really, really compact right before cell division to the structure that you guys are familiar with called a chromosome. And so it does coil, and that degree of coiling is going to make a big difference here. If it's coiled very loosely, you can read the genes, you can see them, you can use them to make your proteins. But if it's coiled up very tight, then you can't see them. You can't see what it says, you can't read it, you can't use it. I always tell the students this is kind of like you went to the bookstore, you bought your book, and it's covered in cellophane, and so if I told you to read what's on page 43, if it's still in the cellophane, you can't read it, right? Whereas if you take the cellophane off, you can open to page 43 and you can read what it says. And so that coiling becomes really important when we're talking about epigenetics also. Okay, so with that as background, what the heck is epigenetics? really started a while ago, a couple of decades ago, and there was problems with getting a definition. And finally, one of these meetings in 2008, this big meeting, they came up with a definition that everybody agreed upon. You'll find out why 2008 makes such a difference to this decision. So, stable, heritable phenotypes resulting from changes in chromosomes without alterations to the DNA sequence. You guys are all familiar with mutations. Mutation, by definition, means that you have changed the DNA itself. What we're saying is that you're changing what the DNA does without changing the DNA itself, right? So this is something that happens outside of or above or upon the DNA that changes what it does, but it doesn't change the DNA itself. So that's epigenetics. Changes can modify the activation of the genes, but not the sequence of the DNA. What this is kind of like, if you think about writing a whole paragraph out, and then if you were to leave the letters in the same order, but change the spacing and the punctuation, it would mean something totally different. Letters are all still there, still in the same order, but the meaning isn't the same. Right? So another important thing, and this became very important later on, is that the changes that are being made outside the DNA or on the DNA have to be preserved when the cells divide. And so there's going to be lots of mechanisms that are being explored right now, and there's questions about, are they really epigenetic? Because you have to be able to show that those changes are going to be inherited in the cells once that cell divides. Let's see, if they occur in the sperm or the egg, and though that lucky sperm and egg meet up and fertilize, those changes can be inherited by the offspring. And so that makes for very interesting consequences also. And some of the factors that do cause epigenetic changes that we can document, and I'm about to show you evidences of, include diet, toxins, and life experiences. Sitting in this room right now hearing this, changing the epigenetics of your genome. Okay, kind of the dawn of this whole thing, where did it start? Back way 1970s, they're working on developmental biology. Not epigeneticists, developmental biologists are working on how do stem cells differentiate? So you've got this stem cell that can go on and become all of the different kinds of cells. And so how do cells become one type of a cell and not other types of the cells? And that's when they discovered methylation and the fact that methylation turns genes off or silences genes. And that was kind of the whole start. They didn't know that they were doing epigenetics, but that's what it turns out they were actually doing. And so this is a great little slide that shows some of the epigenetic mechanisms, and there are a lot of them being investigated, but there are a couple of them that have been investigated extensively. And so one of them is methylation. So if you take your DNA and you put methyl groups all over your DNA, it causes it to coil much tighter so that the genes can't be read. So in other words, you're turning the genes off, right? Haven't changed the DNA, but you're turning the DNA off. The other thing is acetylation, because the histones have little tails on them. If you stick acetyl on them, it actually makes the DNA coil less tight, and so the genes are available, it turns genes on. Those things have been shown to be preserved when cells divide. So the cells that had those traits originally, now the daughter cells will also have the same epigenetic markers on them. And some of the other things that are being investigated are listed down here. That methylation is cool. So back to our DNA, and so here's our base pairs, and it's this cytosine right here that gets methylated. These little enzymes that do this job, hot topic in biology right now, because they want to know what they are responding to when they add those methyl groups. And so there's a whole family of them, thank goodness, just numbered 1, 2, 3, 4. And so they add this methyl group, and then that's going to change the structure or what the DNA is doing. It's kind of interesting, in mammals, what happens is that those methyl groups get put primarily on what we call the promoter region. So here's your section of DNA, that's the genes, that give you information about a particular trait. And upstream, at this end, where this reading would start, is what we call the promoter region. So if you stick all those methyl groups on there, it tightly coils, it can't be read. So in mammals, that's primarily the area that's gonna get methylated to shut the genes off, okay? All right, so some evidence is that this is real. I mean, this occurs, right? And Karen, I'll bet you're familiar with this guy and his experiments, right? Okay, so this was an interesting experiment about your environment interacting with your genetics to give you specific phenotypes. And so this researcher had these great little mice over here, and they were very attentive mothers, and so they fed their young and cuddled them and licked them and everything, provided this nice little home for them. And these mice babies grew up, they were very low stress, happy little rats, okay? And then we've got this mother over here who's not good, and she doesn't suckle very often, it doesn't lick them or anything, and they turn out to be these nervous rats. And so the researchers then dissected their brains, and they took a look at where the methylation patterns in the genes in there. And what they found was that in these guys that were so stressed, there were areas in the brain that are related to a specific receptor that were really heavily methylated. So you have a hormone that's involved in stress response. You have to have a receptor so that the hormone can do its job. Well, if you take the receptor away, the hormone can't do its job. So these little stressed out rats, their gene for the receptor was methylated. They didn't make the receptor. Couldn't get the hormone, couldn't deal with the stress, ended up these nervous little rats, right? And then on the other hand, these guys were at very low methylation. Well, that's interesting, but you need to show whether that's just a genetic trait or not. You know, maybe the one strain had, the one and not the other. So what they did was switch the pups, and so still the same types of rats. Here we got this nice little mother. Now they've switched these rats. She's taking care of these guys. Guess what? They grew up low stress, low methylation. And these guys, she's not taking care of, high stress, high methylation. It showed that that particular trait was not genetic. Okay, so it was not the DNA itself that was causing that. It was something in the environment that was causing that particular phenotype. This was the best part. So, take this inattentive mother, wasn't taking care of the pups, ended up with these high stress ones, and they took this chemical that removes methyl groups, injected it right into the hippocampus. The methyl groups left the DNA, and the pups immediately changed their phenotype. They became very low stressed when it changed them. So that tells you that epigenetic traits can be reversed. This is important. We're going to talk about therapies later, so we want to keep in mind that studies have shown, both in rats and in people and mice and everything, that these things can be reversed. Okay, evidence is that it causes from diet. This is my friend journal down here that was publishing these just about the same time that they were doing the sequence, releasing the sequence. He was publishing this. And so these are these mice that have this agouti trait, that's this yellow color. That agouti trait, when it's expressed, is associated with obesity, high blood pressure, diabetes, all sorts of horrible health effects, right? These two mice actually have exactly the same DNA structure. They are clones. These two have exactly the same genetics, right? The DNA is exactly the same. What he's done is he took these mice and he started putting them on a diet that was really high in foods that donate methyl groups. And so in that diet that had all these methyl groups, then these same mice changed phenotype, and so they became darker and thinner and healthier. The important part about that study was he showed that that was then inherited in the offspring. So these mice went on to have mice that looked just like them. These mice went on to have mice that looked just like them. So it was inherited. They showed that these epigenetic changes could be passed to the next generations. Okay. Here's another one that talks about toxins, and so this particular fungicide is not a mutagen. It has never been shown to interact with DNA in the lab, so it does not cause mutations. And yet, if you give male rats, or mothers, this, and then the male rats have very defective testicles, very low fertility rates, and that is seen up to 4 generations after it. And so an epigenetic change that occurs due to an environmental toxin that's passed to the offspring. Okay. Humans, one of the first studies here with humans was done on those monozygotic twins, and so these are one egg, one sperm come together, but forms 2 babies. So they have identical genetic information, at least at first, because then as you start to grow, you develop all these epigenetic changes in your DNA. And so they did find that when you took the pairs that were younger versus the pairs that were older, you started seeing more and more differences. And here's his data showing that. And so these are the DNA fingerprints, and they were looking for methylation patterns here. So these are 3-year-old twins. Here's one of them, here's the other. You can see that you really just can't even tell them apart on a fingerprint. Now you have 50-year-olds and taking a look at their fingerprints, and you can certainly tell them apart. So these changes have accumulated over time. The statistics, numbers of differences in 3-year-old twins, numbers of differences in 50-year-old twins. So lots of changes accumulating in the DNA as you age. Okay. These studies had a lot more to do with environment effects. And so this guy was doing these studies and comparing different groups. He did suicide versus death by other causes. So in the same geographical area, he took people that had committed suicide versus people that had sudden death by other causes. And he looked at the amounts of methylation in the areas of the brain associated with in the hippocampus associated with stress and such. He found that the people with had committed suicide had much higher methylation in those areas than the others. Not only that, but then he went in and he divided the suicides into people that had been abused as children versus people that had not. And the people that had been abused had the highest levels of methylation in those areas. He also did poverty versus wealth, and these studies he did over time, and he found that the only really big significant difference was with childhood economics. So if your economics were different when you were kids, that's when you could see difference. If it changed later on, if he took kids that came from the same economic status and as adults had different economic status, it didn't really show any difference. And then did the old orphanages thing. So you've got the kids raised in the orphanages versus the kids that were raised in home, again in the same geographical area. And the kids that were raised in the orphanages had a lot higher methylation in there. So environment interacting with the DNA and shutting down different genes. Evidences that those can be inherited really come from this great study done in Sweden. This is a little area up here in Sweden where back 200 years ago there was no way to get to it in winter. And so if they had good harvest, everybody ate really well. If they had bad harvest, everybody starved, right? And so these scientists went back through the records and they were looking at years that were good, and then the health in the coming years, years that were bad, and the health in the coming years. And what they showed very conclusively was that in boys in particular, if they did not eat well in those years right before puberty, it turned out that their sons and grandsons would be very healthy. They'd have low body fat, they'd have low rates of diabetes, low rates of cardiovascular disease. So it was definitely passed through a couple of generations, just caused by dietary changes. Okay, I don't know if you know about genomic imprinting. This is something that's kind of new and I thought was just really fascinating. We are diploids. That means that you get 2 copies of each gene. So we have 46 chromosomes total, and so you have 23 pairs of chromosomes. So you've got 2 of the number 1 chromosome, 2 of the number 2, 2 of the number 3. One of them you got in the egg, one of them you got in the sperm, right? And so if you have 2 of the same chromosome, one from mom, one from dad, that means that you have 2 of each gene, right? So you've got a gene for how to make insulin from mom and a gene for how to make insulin from dad on the homologous chromosome, right? Okay, except for in imprinting, what happens is that we get some permanent shutdown in very specific genes in the genome, and so you only have 1 functional copy of those genes. You can imagine then that if something happens to that 1 functional copy, you don't have any way to compensate. If you get 2 copies of how to make insulin and 1 goes bad, you still got 1 to rely on, right? But if you only got 1 to start with and something goes bad, you got no way to compensate for that. And so here's how basically how imprinting works. And so at one time, this was you, right? So here was the egg from mom, and it's got 1 copy of, say this is chromosome number 1. And here's the sperm from dad, and here's his chromosome number 1. And he's got these epigenetic markers, and she's got these other epigenetic markers on her information, right? Egg and sperm come together, here you are, voila, you're born, right? And so you both have this copy of this information that came from mom with its markers, the copy that came from dad with its markers. So you both have the same, but when you go to make eggs and sperm, so you're going to do this process of mitosis now. And so in the ovaries of gal, so here's the 2 chromosomes she's got for the, say it's chromosome number 1. As part of the meiosis process, it strips all of those markers off of there. So the genetic those markers are gone. And then look what happens. She's going to re-imprint, and she's going to make it match the maternal chromosome. So that now both of her chromosomes are going to say what the mothers originally said. On the other hand, if this is happening in the testicles, and so we're going to be making sperm, we're going to strip it, and then we're going to end up re-imprinting to exactly what dad's chromosome said. So these are going to be then areas that are going to be shut down. They're not going to be functional genes. It's kind of interesting, they've done genome studies for all sorts of species, right? The only organisms that do this are mammals and flowering plants. Everything else that they've investigated, sexually reproducing things, when they do this part right here, they leave them stripped. We're the only ones that preserve those imprinting patterns. And it can have a big effect. So if you're taking a look at this and you're going, if you know classical genetics, right? Dominant, recessive. So if she's got a dominant A and he's got a recessive, she's got a dominant B, he's got a recessive, she's got a recessive C, he's got a dominant C, right? Suppose that imprinting shuts this one down for her and that one down for him. Baby gets this, and whereas you would predict that this offspring would show the dominant A, the dominant B, and the dominant C, because these have been imprinted and shut down, it's not what their phenotype will be. Their phenotype's going to be the dominant A, the recessive B, the recessive C instead. And so, let's see where that would be a problem and could lead to some genetic disorders, right? And so here's examples of genetic disorders. So this is chromosome number 15, and so we're on the long arm of chromosome number 15. There's this little section right in here that does have imprinting on it Females will have a couple of genes imprinted there. Males will have at least one gene imprinted on that little section. And so in that section, there are several genes that you only get one functional copy of a particular gene, right? And so you can see if anything goes wrong with your one functional copy, you're just out of luck, right? And so there are typical diseases, and the typical genetics, what usually happens is that you end up with a deletion or a non-disjunction or a gene mutation. Those are common, regular genetic disorders, right? And they result in this, but now they've also shown that it can just be an imprinting error, so leaving you no functional copies of this. And if this happens on the paternal side, you're going to end up with a disease called PWS, and you're going to have one particular set of phenotypes. Same thing that can happen for the other one, and you can also have an imprinting error. And so if it happens on the maternal side, whole different set of symptoms. So same area affected, can be by just straight epigenetic mechanisms, and you end up with some pretty severe genetic disorders. Here's probably what's most interesting to you and applicable to you. This applies to cancer. You know how many people have cancer or will come down with cancer at some point in their life, right? And so epigeneticists have been taking a closer look at this, and they're seeing a lot more relationships to epigenetics and cancer. And in fact, they found that there are imprinted genes, there are imprinted genes from mom that usually would normally suppress cell growth, and there's imprinted genes from dad that would stimulate cell growth. Cancers are all about cell growth. This is the normal cell cycle, and so normally the cell cycle has to be tightly regulated. So if you're a skin cell or something that's supposed to divide fairly rapidly, it's going to be a fairly rapid cycle for you, days or whatever, right? But if you're a liver cell or something that's going to reproduce slower, then it has to be a very, very slow cycle. So to keep the right number of body cells, it's all about controlling the cell cycle. If you lose control of that cell cycle, cells start being made too quickly, you end up with masses of cells, tumors, and tumors can lead to cancer, right? And so it's important that we've got control of that, and yet they're finding that some of these imprinted genes are involved with the regulating that growth. And so they have seen that some of the tumor suppressing genes that are maternally expressed mistakenly turned off. So now my things that are supposed to suppress the cell cycle, I turn off. And so my cycle's going too fast and I'm making too many cells. Same thing with the paternal ones. So a lot of these are now associated with oncogenes. You're probably familiar with that term. And so again, losing control of that cell cycle due to changes in these imprinted genes. There's a lot of different cancers that have now been associated with this. This is just a small sampling of some of the most common ones. And so imprinting problems and ending up with cancers from that. Okay, so some applications. So I don't know if you've seen any of those articles yet or heard any of those little commercials where the pharmaceutical companies, the biotech companies, we're working on epigenetic therapies. We're going to be able to fix everything, right? So they're going to fix boost learning and memory. They're going to treat depression. They're going to just cure all type of things, right? Is any of that reality? Yes, some of it actually is reality. There was an article published last year that in osteoarthritis, they found this very important epigenetic mechanism in an enzyme there. And so if they can change the methylation pattern for the gene for that particular enzyme, they may be able to cure osteoarthritis. And so that's actually being worked on. Or closer to home, right down here at MD Anderson, one of our very famous cancer researchers, now thinks that lots of the cancers, particularly the cancers that are known to interact with the environment, so environmental exposures, diet exposures, toxin exposures, you end up with cancers. He's thinking that most of these are probably going to turn out to be epigenetic changes, which to him is good news because he can reverse epigenetic changes. You can't really reverse mutations, but you can reverse the epigenetics. And in fact, he's working on a leukemia epigenetic therapy right now. So, woohoo! Right? Okay, so in 2008, that's why 2008 became an important year for epigenetics. They had to come up with a definition of what it was because the NIH was releasing these funds for the next 5 years, $190 million just to fund epigenetic studies. And so when they announced that, they said that it's going to have potential to explain mechanisms in aging. I hope so. Human development, origins of cancer, heart disease, and they do think that it's going to turn out to be really, really significant. So just want to link you to this real short, just because you take a look at this huge meeting that's coming up in 2 weeks. I think my dean should send it to me.
Yeah.
So this is the NIH's epigenetic site, and they do have a major meeting coming up just in 2 weeks. And so since they started funding these things in 2009, this would be about the time when all the really great stuff starts hitting the press, and so it would be a great thing to go to. Hint.
Don't even let you
And if you wanted to know more, a friend of mine has an acquaintance who took the course at Berkeley, said it's absolutely fantastic. If you want to hear more from Randy Dirtle, he does a really great podcast. He just retired, sadly. So he's got a great podcast though on the NIH site. And there was 2 books that were just released last year. I haven't read them cover to cover, but I've cruised through them, and they both look like they're pretty darn good. I wouldn't get anything on epigenetics that's more than a couple of years old because it's changed so much. So take-home messages. You can't change the DNA that you inherited, but you can certainly change the epigenetics, right? And if you plan to have kids, everything that you're doing today can have an effect on your kids and your grandkids. So this poor little gal down here is trying to decide my carrots and cake, right? And in the future, I do think that you're going to see relatively soon, you're going to find epigenetic therapies and some huge changes, particularly in cancer and autoimmune diseases. That's it. What questions do you have?